Hi, everyone. Welcome to the 2022 Gladys J. Everson Lectureship in Biochemistry. So first, I just wanted to say a few words of introduction about Gladys J. Everson. So she is an alumna of the Department of Biochemistry. She received her PhD in 1942 under the mentorship of Harry Steenbach. And her research interests were in the relationship between dietary deficiencies and birth defects. So with that type of research program, she went on to join the faculty at UC Davis and had a long career there, eventually rising through the ranks to become department chair, where she promoted the need for better exercise and nutrition. And she later bequeathed her estate to the Department of Biochemistry to support the Gladys J. Everson Lectureship in Biochemistry, which allows us to invite back distinguished alumni and affiliates of the Department of Biochemistry. So this year, we're fortunate to have Dustin Maly as our Everson lecturer. So Dusty was an undergraduate here at the University of Wisconsin, and he actually earned his degree in chemistry, and we'll learn a little bit more about that later. He was an undergraduate researcher with Howard Zimmerman, and then Dave Brow, and then Laura Kiesling. And in the Kiesling lab was where he really shone. He told me that he was a top-tier graduate student there, and he was actually invited to Laura and Ron's wedding. That's how great he was in the lab, so really S-tier student. He then went on to UC Berkeley for his PhD, where he worked under the mentorship of John Ellman to develop molecules that selectively bind to other molecules using chemical and chemical biology tools. And then he did postdoctoral training with Kayvon Shokat at UCSF, where he used chemical biology to define kinase substrate pairs. So you may notice that Dusty got his PhD in 2002, but if you look at his CV, it says that he graduated from Wisconsin in 1999. So is he really so great that he only took 3 years to get his PhD? Well, it turns out that he left Wisconsin in 1997, not really realizing or registering, I guess, that he hadn't actually graduated. So it was only after he passed his qualifying exam in John Ellman's lab that Berkeley actually checked to see if he had an undergraduate degree, and he didn't. So he took a correspondence French class through Wisconsin and finally was awarded his degree in 1999. So it all worked out. And now he is on the faculty at the University of Washington, where his research program is focused around using chemical biology to understand cellular signaling. And for this research, which we'll hear about today, he has been recognized with numerous awards, including the Sloan Fellowship, the Camille Dreyfus Teacher Scholar Award, and the NSF Career Award. So without further ado, I'll let Dusty take it away.
Thanks a lot. Thank you very much. So 2 pieces of advice for people. If you're talking to your host, tell them stories about you after your seminar. And the other is, I guess this is more of some information. Please don't ask me to speak any French. I only do it through the mail. So, but yeah, thank you very much for the introduction. And actually, all of what Amy said is true. And what I'd like to, it's actually a great honor to be here and give this talk. But before I do, I sort of want to just acknowledge some people and my great time here, despite Wisconsin being very particular about the requirements for graduation. It was actually a really fantastic experience, and I can really thank 2 people. There's many people I could thank, but 2 people in particular I really like to thank, and one's Dave Brow, who's here, and it's great to see him. The time that I spent in his lab was really what got me very excited about research. And the thing that was really great about it is, despite I wasn't the most productive undergrad in his lab, but he was very patient and was very willing to humor my enthusiasm, and I really appreciate that. And really, that experience is what inspired me to really want to be a scientist. And the other person I'd like to thank is Laura, who's not here anymore, both for, again, giving me a great research experience in chemical biology, and also for really making it so that I could withstand pretty much any mockery that could be thrown at me. And it builds character. But despite that, she's been incredibly supportive throughout my career. And so as Amy mentioned, I spent my time here from 1992 to 1997 before graduating in 1999. And and I think from that, I thought prior to this lectureship, the highlight of my career was I was actually featured in the 2009 Biochemistry newsletter. So this will top that experience. So before, I had to get a little nostalgic and just mention one more thing. I was thinking as I was coming here what things I like to do besides academics and research. So I thought about it. I really like to go to the exclusive company, and at the time it was the 90s. So I'd buy some vinyl, some grunge vinyl. Unfortunately, the exclusive company's not around anymore. I would like to go to Gumby's, which has some of the most disgusting but the least expensive pizza you'll ever eat. But unfortunately, Gumby's is no longer around. But the two things that are around that I really enjoyed are the Terrace, as well as I recommend to anyone the Hoofers, which is a great organization where I learned a lot about doing things in the outdoor, which I sort of carried out west. So now that you've all indulged me in taking a trip down memory lane, I'll tell you about the topic that is the title of my talk. And that's the idea of how we can allosterically modulate protein kinases, both understand this allosteric modulation, but also use some tricks that we can, as chemists, for sort of modulating these through allosteric, through the ATP binding site. So I'm going to tell you about that today, but before I sort of lay out this concept and how we do it, I'd first like to take a step back and just tell you a little bit about protein kinases and why what I'm talking or what I've proposed at the outset seems reasonable. So just as some background here, protein kinases are an enzyme family. There are 538 of these in the human genome. This is a commonly used graphic. It's a dendrogram of all the kinases in the human kinome. And what all of these do, except for the small percentage that are pseudokinases or catalytically inactive, but all these do is catalyze the transfer of the gamma phosphate of ATP to a serine 3 or tyrosine residue. And what we see is that these kinases, despite containing the same and very similar catalytic domain that looks sort of like this bean-shaped three-dimensional structure with a cleft in the middle where ATP binding, where ATP interacts, play really diverse roles within the cell. And the way that kinases play diverse roles within the cell is that there's two aspects of this. One is that they're highly regulated. So many kinases either have sites of phosphorylation or external regulatory domains that regulate their activity. So there's high temporal and spatial precision in their regulation. And the other thing that kinases share is that these surfaces that regulate their activity also serve other roles in signaling, and these are what we call phosphotransferase independent functions. So many of these kinases on binding surfaces adjacent to the ATP binding site will bind to other enzymes and activate them or inactivate them. They can often serve as scaffolds where they'll bring together a number of signaling enzymes. So the bottom line here is that kinases are able to obtain highly diverse function by coupling their ability to phosphorylate things and with high spatial temporal precision with their ability to do other things beyond just put phosphate on other proteins. And so one thing when I started out that we were really curious about is we were wondering if it would be possible to modulate these functions, these other functions beyond phosphate transfer and the phosphotransfer activity through the ATP binding site of kinases. So would we be able to take and modulate the ability of a kinase to, say, activate another enzyme or to scaffold and potentially do this divergently depending upon how we engage the ATP binding site, but could we do this through the ATP binding site? And the reason we thought this might be possible is if we take and we think about this catalytic domain that has this ATP binding site, is that there's two things that over the last few decades that have emerged. One is that the active sites, the ATP binding sites of kinases, and this is the three crystal structures of the same kinase, are highly dynamic. they can undergo large conformational changes. And what we now know is that most kinases are able to adopt at least 3 different conformations that have very different orientations of key catalytic residues. So one of which I'll talk about is the active conformation, in which all the residues are aligned for catalysis. There's also 2 inactive conformations that are commonly observed. One is where the activation loop, which is a signature of kinase activation, undergoes a large conformational change, creating a hydrophobic pocket. And the third is one where a key helix, which I'll talk about quite a bit, called helix alpha C, undergoes a large rotation where a glutamic acid that's important for catalysis is moved out of the position for undergoing or catalyzing phosphate transfer. And so, as I said, this is one thing we knew, that the same kinase can adopt these different conformations that are very different structurally. But the other thing that was observed early on is that we can identify small molecules that bind to the ATP binding site with high potency, and as I'll talk about with enough work, reasonable selectivity. But we can also, based upon the same scaffold, identify ligands that stabilize those 3 conformations that I showed you. So all of these ligands I'm showing here have in gray moieties that look like the adenine ring of ATP, but depending upon what substituents they project into the ATP binding site, they either can stabilize the active conformation, this conformation where the activation loop is flipped, or the third conformation where this helix is rotated out. And so what we speculated is that, based upon the fact that these regulatory domains, right, that I talked about that regulate kinase activity, communicate with the ATP binding site, that we would be able to take and, by understanding how these regulatory domains affect the conformation of the ATP binding site. So this is an example where the regulatory domains are stabilizing an active conformation, but when they undergo a conformational change, they stabilize this inactive conformation. By understanding these relationships and then using ligands that stabilize a specific conformation, we could actually promote disengagement or engagement of these regulatory domains and surfaces. And by doing so, take the same kinase and either promote or inhibit these interactions or these functions that are independent of its ability to phosphorylate things. So for example, here's a kinase that plays a role in scaffolding. Here's a ligand that stabilizes the ATP binding site in an active conformation. And by adding this inhibitor, we would inhibit its kinase activity or its ability to phosphorylate things, but promote its scaffolding function. And vice versa, if we had an inhibitor that, in this case, recognized the kinase domain of the active or the inactive conformation, it could actually promote disassembly of this scaffolding and stabilize the monomeric kinase. So that's sort of the concept. And really, if we think about this from more of a drug discovery standpoint, it becomes a question or a hypothesis that we had. And the idea was that we could take the same protein kinase, we could have inhibitors that are highly selective for that kinase, and depending upon, and they would both target the same site, but depending upon the interactions they make in the ATP binding site and the conformation they stabilize, they could actually give you a differential phenotypic effect, right? And that would be based upon these phosphotransferase independent functions. So that's our hypothesis, and this is what my lab has been looking at for at least a decade. And what I'll tell you today are about 2 stories. One will be a longer, fuller story. One will be a bit shorter. The first of which is a story I'll talk about this ER-resident enzyme, or a kinase, called IRE1 alpha. It's a kinase that sits in the ER membrane and senses ER stress. And I'll tell you about how we can think about its regulation and its modulation. And the second story I'll tell you about is Src kinase, a Src tyrosine kinase. It's an enzyme that plays a role in multiple signaling processes, including migration, integrin signaling, and other diverse processes. So that's the plan. All right. So the first story I tell you about, as I mentioned, is IRE1 alpha, right? So IRE1 alpha is highly conserved from yeast to humans. In yeast to humans, it shares a very similar architecture. As I mentioned, it sits in the ER. It presents a luminal domain, which sits in the lumen of the ER, and a module that's connected through a transmembrane region and a linker that's connected to a kinase and an RNase. And what we know about IRE1 in both yeast and humans is that what it does is serve as a sensor of stress in the ER. And so, just a quick overview, right? Proteins that either are secreted or end up as transmembrane proteins, right, go through the ER, where they're basically transported, and often, right, especially for secreted proteins, there's a series of disulfide formation, folding, and other processing enzymes that ensure proper folding of proteins that are secreted or end up at the plasma membrane. What is known is, especially for cells, as I'll talk about, say, like the pancreatic islets or B cells that secrete large amounts of proteins, it's possible that the folding capacity of the ER does not keep up with the ability for the ER to process it. And what happens is, under these ER stress conditions, IRE1 is activated, and essentially we have a series of trans-autophosphorylation in the cytosolic domain, which leads to the RNAs being activated, the cleavage of a messenger RNA called XBP1, which then is ligated by a RNA ligase, RICB, and leads to a series of transcriptional events which lead to, especially in yeast, the transcription of oxoreductases, chaperones, and things that can help increase the folding capacity. So that's IRE1. I'd like to, before I go into this, sort of acknowledge my long-time collaborator, Feroz Papa at UCSF, who got me interested in IRE1 and thinking about how we can modulate it allosterically. All right, so that's an introduction. What we were very curious about is whether we could take and target the kinase domain, the ATP binding site of IRE1, and control its RNase activity. And the thing that got us most interested in this is a study that Feroz did when he was a postdoc in Peter Walter's lab. And what Feroz was set out to do is actually to try to take and use a bump hole strategy that was developed by my postdoctoral mentor, Keyvan Shokat, and use that to label the substrates of the kinase domain. It turned out, though, when he introduced this point mutant, introduced this hole into IRE1, it didn't allow him to label the substrates because the kinase was completely dead. Okay, so it was basically a failed experiment. This is something, after 20 years, I still haven't asked Feroz why he did this, but what he decided to do, which ended up being providing a lot of insight, is he said, well, I have this, my mutant IRE1 being expressed in yeast. It's essentially totally catalytically inactive, but Keyvan not only has a system where he has these bumped ATP analogs, but he has these selective inhibitors. I'm going to basically take an inhibitor and I'm going to add it to this crippled kinase. And what he saw was pretty remarkable. When he added this inhibitor of an enzyme that's dead, the RNase turned on. Okay, so very counterintuitive, but it was very informative because of what it told us, right, is that the kinase and the RNase domain are in some sort of communication, and that you don't need this trans-autophosphorylation or the ability of IRE1 itself to phosphorylate anything to actually activate the RNase, right? And for this reason, we and other people, including Peter and Keyvan, set out to try to understand this allosteric relationship and whether we could control this with small molecules. Okay, so to do this, I guess I'm a chemist / biochemist. In trying to understand this, we want to use a system that's as simplified as possible. What we ended up doing to originally sort of attack this problem is we did some biochemistry on IRE1 that just contains the cytosolic region, the kinase and RNase domain. We aren't using the transmembrane domain or the luminal domain. and we performed a series of enzymatic assays. So I'll be telling you about 2 of these today. One is just a standard kinase assay. And this is a nice assay because what we can do is we can probe the activity of IRE1, its ability to phosphorylate, and we can determine whether these molecules that I'll talk about, these ATP binding site ligands, occupy the ATP binding site of IRE1 alpha by determining whether we inhibit this kinase reaction. So this is a way of determining ATP binding site occupancy. The other assay we have is an RNase assay in which we can take our purified IRE1 alpha, this is just the cytosolic region, use a quenched fluorogenic substrate, and measure the RNase activity of this. We first can set out to understand a little bit about how, in this case, phosphorylation in this purified constructs affects IRE1 alpha's RNase activity. We can take a construct that we've purified from insect cells that's highly autophosphorylated. We can assay that, and we can compare that to a version in which we've dephosphorylated this construct. We can then do an assay where we add increasing amounts of the enzyme, and we determine what the RNase activity looks like. And what we see, and this we can fit this in terms of a curve, right, is that IRE1 alpha must form at least a dimer. The Hill coefficient's a bit steeper, so it looks like it might form a tetramer, closer to a tetramer, but this dimer/oligomer formation is necessary for RNase activity. We can also fit this, right, where we actually get a K dimer, so the affinity of the protomers for each other, and what you can see is the purified construct itself is submicromolar, so about 150 nanomolar. If we compare that now to the unphosphorylated form, what we see is that the K dimer is much lower. We actually can't generate enough enzyme, get it to high enough concentration that we actually can fit a K dimer, but it essentially tells us what the difference in the ability of this to dimerize and form the RNase active dimer is based upon concentration. All right, so what we set out to do is to try to understand Feroze's result, and I'll be honest here, I'm gonna tell you a story. It's gonna be very pretty linear. This is not linear in terms of time, but hopefully it'll be as clear as possible. But what we set out to do is try to understand is, you know, for IRE1, is the relationship between occupation of the ATP binding site and RNase activity, is that something that is true just for this weird sort of mutant that's broken, or is this a common feature? And so what we set out to do is we used our kinase assay. We have, you know, within the lab, libraries that we can purchase of kinase inhibitors, ATP binding site directed kinase inhibitors, and we just did a screen. We just found everything that bound and inhibited the ability of IRE1 to phosphorylate something, so things that occupy the ATP binding site. So we were able to come up with a pretty diverse set of inhibitors, and then we asked ourselves, okay, how do these affect the RNase activity of IRE1 alpha? So in this case, I'm just showing the results here. This is the enzymatic activity of this dephosphorylated construct. I showed you that before, right? It has a low propensity to dimerize. In this case, we're adding inhibitor to saturating concentration, or as close as we can get, and we're looking to see what is the RNase activity when this dephosphorylated form is bound to the inhibitor. And what we saw is in every single case, these inhibitors activated our IRE1's RNase activity. Okay? And if we went and we did this titration in terms of now taking and generating the IRE1, in this case, dephosphorylated IRE1 alpha inhibitor complex, and then determining how this affects K dimer, is what we see is all of these inhibitors essentially activate the RNase by promoting dimer formation. So we see this relationship in the wild type enzyme that correlated to this mutant in yeast that Feroze initially characterized. Okay? And so, you know, here's just a quick model of what we think, right? That basically either the unphosphorylated form, which would be present in the absence of ER stress, or this phosphorylated form, which forms in the presence. But basically, the both forms of IRE1, ligands that occupy the ATP binding site will prevent dimer formation and which prevents RNA, or promotes RNase activity. But one question we had is, can we break this relationship? Is it possible to identify ligands that bind to the same site, but rather than promoting dimer, and promoting RNA's activity, can we find something that does not? Okay. First of all, before I tell you how we might go about that, what's the motivation for this? Well, I told you and I gave you a picture of what IRE1 alpha does. And what it does is provide an adaptive response to cells that are undergoing ER stress, right? Which we call this adaptive response. And that's in response, or essentially generation of this spliced XPP1 transcription factor. But for mammals, there's a dark side to IRE1 alpha, that when cells are undergoing very extreme, often irremediable ER stress, the adaptive UPR response is actually overwhelmed by a terminal UPR response, right? And essentially what it means is the cells, and this happens in cells that have a high load in terms of secretion, we often see this, they basically reach a point where they're overwhelmed and they undergo programmed apoptosis. And what this terminal UPR is characterized is by increase in this protein called TIC-SNP. It involves caspase-mediated apoptosis, as well as something I will talk about where large amounts of messenger RNAs undergo decay, which is called RID. So these are all things that, in general, cause the cell, right, that's under this severe ER stress to undergo programmed cell death. And so that was our motivation. We were curious, and especially for diseases of cell degeneration, so diabetes, I'll talk about it a little bit for this, where there's this signature of severe ER stress. We were just motivated to say, like, well, can we develop molecules that will actually turn off IRE1 to see if by turning off IRE1 through the ATP binding site, this will allow us to see if this will help cells that are overwhelmed with ER stress to promote pro-survival. All right, so we set out to do that. I showed you before, right, these the different conformations that the ATP binding sites of many kinases can adopt. What we did is we took a targeted approach where we took those two inactive forms I mentioned, one where we flipped the activation loop, one where we moved the helix alpha C. We made a lot of inhibitors. We thought, okay, well, probably stabilizing one of those conformations will be the key. If it's possible to turn off the RNAs through the ATP binding site, we'll probably want to use one of those motifs. We screened a lot of molecules, and Guiney, the graduate student at the time who did this, made a lot of molecules, and it was very, very difficult. But finally, in doing this screen, we got one hit. I made this very small so you can't see it because it's embarrassing, but it was essentially a 60 micromolar hit. Anyone who is doing inhibitor discovery, small molecule inhibitor discovery, well, we usually say if someone comes with a 60 micromolar inhibitor, they'll say go away because it's not great. But it was the only hit we had. And so basically by having this, we could sort of focus in, and this has been many years now, but basically by looking at this motif and optimizing it, we've now been able to take and identify inhibitors. So this is our initial set, this inhibitor Kira6 that we generated. Amgen developed, based upon our work, developed an even better, more potent and selective inhibitor. And now we can generate inhibitors that are single-digit nanomolar for inhibiting the kinase, right? Because they're kinase inhibitors, but also inhibiting the RNAs by performing this RNase inhibition screen. So that's what we had. So how do these work? How do these inhibitors that we've identified now that are much more potent, how are they able to turn off the RNAs? Okay, and so we can go back to our enzymatic assays and determine how these molecules are able to inhibit the RNA's activity. So the first thing we can do is just do a comparison where we titrate our concentration of IRE1 alpha. In this case, we have the phosphorylated form, which has a higher propensity to form dimers. I showed you this before. This has a K dimer of about 150 nanomolar. We can then do the same titration in which now we add a saturating amount of our inhibitor, our kinase inhibiting RNase attenuator, we call them Kiras, and do the same titration. And what we see is at no concentration, we can only reach about 2 micromolar of the enzyme, do we see any RNase activity, okay? Which we interpret as the inhibitor stabilizing a conformation that prevents dimer formation, right? And we've gone a little higher, and we never are able to recover RNase activity. And we can also do other sort of crude assays to get at this. This is a non-specific cross-linking assay where we're just taking increasing concentrations of iruron alpha. We then add disuccinyl superate, so a non-specific lysine cross-linker. If we do this with the APO enzyme, in which we see an increase in RNase activity, we can see at fairly low concentrations formation of the dimer and a ligament from which we interpret as that back-to-back dimer I showed you. And in the presence of our Kira, we already see a little bit of dimer or ligament, but we almost completely suppress it. So what we think is that these inhibitors are basically inhibiting the RNase by preventing the dimer and stabilizing the monomeric form. All right, I'll show you a little more on how we think this is occurring, right? Because right now we have a biochemical result, right? We when IRE1 is bound to this inhibitor, its RNA is inactive. It's much less likely to form dimers based upon our cross-linking and a few other assays. So I'll tell you how we think we do that shortly. But first, everything I've shown you so far is with a purified construct that just contains the cytosolic domain. So how does this affect cellular IRE1? Well, here's some results that are confirmatory with our biochemical results. We can take IRE1 alpha, that is a GFP fusion. We can add, in this case, ER stress, which causes IRE1 to undergo trans-autophosphorylation oligomerization. And if we take those same cells in which we add ER stress, but we pre-treat with a Kira, we prevent this oligomer formation. So at least, this is not, we aren't able to actually see dimers or specific oligomeric states, but at least we can prevent, right, this higher order oligomer formation that we see under ER stress. We also see that these Kira molecules block the ability of IRE1 alpha to splice or to cleave XBP1, which leads to the splice product. So if we do, in this case, an assay where we look for the splice form of XBP1, in this case, in a system in which we overexpress IRE1 to drive the activity, both the adaptive and the terminal outputs of IRE1, we can see that in the presence of one of our Kiras, or many of our Kiras that we've tested, that we can block the ability of IRE1 to generate spliced XPP1. Not only do we block the adaptive outputs of IRE1, but we can also block these terminal. So these are two substrates, which are these RID substrates, these ER-localized mRNA decay. And in this overexpression system, by overexpressing IRE1, we see depletion both of insulin-1 and BLOS1 in the cell line, which are these on the messenger RNA level. And by treating with our Kira, we basically preserve the messenger RNA. And in terms of functional level, we see that in this IRE1 overexpression system, in these INS1 cells, we see an increase in apoptosis, which we're seeing by annexin staining. And by pre-treating with a Kira, we largely blunt the annexin response, or the annexin staining. All right, so that's on a functional level, right? We're able to block both the adaptive and the terminal outputs of IRE1 with these Kira molecules, which in this case, at least in this overexpression system, as well as in cultured cells in which we add ER stress, basically prevents cell death. And so over the years, as we've generated more selective and potent molecules, some of which I showed you, one of which we've heavily relied on is this molecule developed by Amgen, we've been able to use these now in vivo studies. We're very interested in targeting IRE1 in both type 1 and type 2 diabetes. And I won't go into this too much, but I'm happy to answer questions if people are interested. But in multiple mouse models, we've shown that we can basically, in this case, this is a keto model, a keto model of diabetes in which there's a mutation in insulin 2 in mice. We can basically, by administering one of our Kiras, we can prevent the hyperglycemia that forms. And in this type 1 diabetes mouse model, the NOD mouse model, we can actually reverse type 1 diabetes with our Kira. And like I said, I won't go into this too much because this is, if you're interested, this is published data. But or I'd have to answer questions, but it basically shows that our motivation, or at least partially is something that shows that our motivation for trying to blunt the RNA's activity of IRE1 was justified. All right, so that's where we were for quite a while. But one thing that we got a lot of questions about, and one thing that was always a concern, is that when we use our molecules, our KIRAs, right, our molecules that occupy the ATP binding site of IRE1 and inhibit the RNAs, we're basically both turning off the terminal UPR, which we want to do, but we're also inhibiting this adaptive UPR. And as I showed you in some of these diabetic mouse models, it turned out to be efficacious enough that essentially you still saw a positive effect. But there's still questions about, well, if you're systemically, right, turning off IRE1, which you would be in a orally available drug, is the fact that you're turning off both of these arms of IRE1's output, is that going to be detrimental? And the thing is, at the time, we just didn't have the tools to answer this, right? We had one tool where we knew we can turn on, or we see IRE1 being active. We have this other tool where we can turn it completely off. But we were never able to really try to separate these outputs and determine if we could turn the terminal off and keep the adaptive, would that be beneficial? So that's what we set out to do. This is the work of a super talented graduate student named Hannah Feldman. And what she set out to do is answer first, can we develop molecules that will essentially allow XPP1 splicing to be maintained, we maintain the adaptive response, but then inhibit the terminal UPR arm of IRE1 alpha. So in order to do this, we sort of went back and said, well, if this is what we'd like to do, how conceptually would we do this, right? So we returned to structural biology, biochemistry, and really trying to understand how these KIRA molecules work. So one thing I'll just add here is that this project has been great, but also in many cases very frustrating. One problem we ran into is that we wanted to obtain high resolution crystal structures of our molecules bound to IRE1. And so we can generate this in decent amounts in insect cells, purify it, and set up co-crystallization small molecule. We did this many, many times, often because IRE1 alpha ligomerizes, it would crash out, and so you lose all of your protein. Another thing we would also do is we'd try to co-crystallize with our small molecule. We'd often just end up with apo IRE1 alpha. We did this many, many times. We have many crystal structures of the active form of IRE1 alpha without any inhibitor. This ended up being, although it was frustrating, it was useful because now we had a really good idea of what the active enzyme looked like, where all the residues are lined. But we were finally able to, both from our work and from other crystal structures, get a good idea of how these KIRA molecules actually affect the ATP binding site of IRE1. And the key here is this region that I'll talk about next couple of slides, called helix alpha C. It's I mentioned it before, it's this region that lines the ATP binding site that rotates out when IRE1 alpha is in the inactive form. And the key to our purposes, it forms part of the dimeric interface between this back-to-back RNA's active dimer. And what we learned is that the key attribute of KIRAs that basically allow them to inhibit the RNAs by stabilizing the monomer is their ability to push this helix alpha C out from an inactive position to an active position. Or sorry, from an active position to an inactive position. All right, so that was our simple model. And what we thought in our hypothesis here, or I guess a prediction, is, well, if we want to basically be able to separate this adaptive from the terminal UPR, rather than having molecules that completely push this helix into a conformation where you can't form dimer, maybe we could identify molecules that only partially displace this, form an intermediate conformation, and allow weak dimer formation, and maybe this would allow us to separate these two outputs. And so to do that, we wanted to go back and analyze the structures of these molecules. I mentioned this molecule from Amgen that was super useful. It turns out for fairly mundane purposes, this scaffold is very useful. The main reason being is that it was a key element where we could have the part that bound and overlapped with adenine and the adenosine ring, it was high enough affinity that we could play around with the region that points towards this helix alpha C pocket, which we think we want to modulate to get this differential effect. And so what we were able to do is use a scaffold now to do a more systematic study and try to understand a few things. One is, when does an ATP binding site ligand go from being, in this case, potentially an activator, to a chira, where we inactivate IRE1, so from activator to inactivator, and can we find somewhere in between? And so these are two molecules that are super informative for this. This molecule in which we just have this aniline being projected in the helix alpha C pocket, we see in our RNase assay activates IRE1. We then compare it to almost an identical molecule. In this case, we only have this sulfonamide, this trifluoromethylpropyl sulfonamide, and basically that acts as a chira. So basically, everything else, it's a multivariable equation. We've isolated the variables. The only difference here is this presence of the sulfonamide or not. We go from activator to inactivator. And this series is nice because now we can do our dimerization. Here we have our chira, which monomerizes IRE1. Here we have our activator. And as we predict, they affect the propensity to form dimer. Okay? So with this series, and I'll show you, I'm going to sort of skip towards slightly different molecules, but what we're able to do is go through and say, well, if we have our molecules that activate IRE1 or our molecules that completely inactivate it, the molecules that partially inactivate, well, the ones that we think might separate these outputs of IRE1, probably lie somewhere in between these two in terms of size. That was a pretty simplistic model. And we were able to identify that. So as I mentioned, the scaffold's slightly different. I've replaced the fluorophenyl, in this case, with this naphtho ring. But we're able to identify two molecules. In this case, we have a chira. It's similar to the molecule I showed you before. It has this trifluoromethyl or trifluoropropyl. But we're able to identify what we call this partial antagonist, which only differs by one methylene. So now if we take these two molecules, one of which we know completely, this chira, which we know I showed you before, a similar molecule, it completely monomerizes IRE1 and inhibits the RNA. We can then do, in this case, this is an RNase inhibition assay. As we see, this molecule inhibits 100% of the RNase activity. But now if we compare this to this molecule that has one methylene difference, we see only partial inhibition of the RNase in this in vitro assay. We can then do our titration, where we now take and compare the IRE1 alpha inhibitor complexes, and we see our chira, as I showed you before. We never see an increase in RNase activity, but these partial antagonists that only separates by one methylene allows some amount of dimer formation. All right, so at least we have molecules now that allow us to test a hypothesis. Right, we have one that is we have two molecules. They are very structurally similar. They have very similar potencies, one of which completely suppresses the RNA activity and stabilizes the monomer, and the other allows some amount of dimer formation, some RNase activity, when there's full occupancy of IRE1 alpha's RNase. All right, so before I get and show you whether these work or not, I just want to show you structurally that these behave as we expect. We're able to obtain a crystal structure of that pair I showed you, this partial agonist I showed you before. Interestingly, if we took a similar chira and we obtain the crystal structure as we'd expect, it crystallizes a monomer. In this case, our pair molecule forms a dimer, but it has if you look at the electron density, that has a partially disrupted dimeric interface. And as we predicted, this molecule displaces helix alpha C from an active conformation. But if we look at this displacement, the displacement is to a lesser degree than a chira displaces the helix alpha C. So it looks like we can find this intermediate range of conformational movement of this helix, which only weakens dimer formation rather than completely suppressing it at full occupancy. And there's different ways of sort of measuring these distances, but we see this overall. All right. So as I mentioned, the nice thing about these molecules is that they have very similar potency. They almost have identical selectivity. This is a kinome profiling method we use where we can profile the selectivity of our inhibitors against about 200 kinases. They're both pretty selective for IR1 alpha. They have a few off targets, but the good thing for us, they're the same off targets. So the differences we see should be due to the common target IR1 alpha that they engage. They also have a similar ability to prevent autophosphorylation of cellular IRE1. In this case, we're just using a phosphate gel to look for inhibition of the autophospholipid form in this IRE1 overexpression line that I told you about. All right, but then the moment of truth is, are we able to differentiate, and in this case, our partial antagonists maintain XBP1 splicing but block RID versus our Kira, which blocks both. All right, and what we see is that this is the case. So here's our splicing assay again. This is, again, a cell line that overexpresses IRE1, which drives XBP1 splicing. If we look at our Kira, this is the data I showed you before. We inhibit the ability to form the spliced XBP1 product, but our pair 1 molecule, which only differs by 1-methylene, is fully permissive of XBP1 splicing. But now if we look at RID, so this is the quantification of the splicing. Or sorry, this is under ER stress. We see the same effect, the ability to permit XBP1 splicing. But now if you look at RID, so this is our BLOS1 substrate, and in both islets and this insulinoma line, we see that both molecules have a similar ability to block RID. So differential effects in XBP1 splicing, the adaptive response, but differential effect on XBP1 splicing, but the same effect on the ability to block RID. And most importantly for us, and this is a case where we used RNA-seq to globally look at this RID effect where we see a depletion of messenger RNA. Both molecules block the RID signature that is induced by overexpression of IRE1, and both, to a similar degree, block the pro-apoptotic, pro-cell death outputs that IRE1 promotes. And this is also seen in terms of annexin staining as well as cleaved caspase. So the bottom line here is that these molecules that have very similar potency and selectivity and only differ by 1-methylene unit, allows this adaptive output within these cellular systems to be maintained, which is different than our Kiras, which block both. All right, so these were nice model molecules for understanding mechanistically what we could do for IRE1. We're really interested in using these in vivo models, especially in diabetes. In order to do that, though, it turns out taking a molecule that is somewhat potent and somewhat selective and turning it into a highly potent, highly selective molecule that has favorable enough pharmacokinetics and ADME properties is pretty challenging, but we've been pretty lucky, and Hannah's led this effort to make some molecules that are as potent and selective as the Kiras we've developed. So now we have 2 molecule sets in our hands that will allow us to actually test this hypothesis in these in vivo efficacy studies, especially in type 1 mouse models of type 1, type 2 diabetes. Is the ability to maintain this adaptive response going to be beneficial? And unfortunately, I don't have the answer to that for you right now, but that's essentially what we'll be pursuing going forward. So that's the IRE1 story. And just to recap in terms of what this means for us broadly is, I was telling you we'd like to explore this idea that we can modulate the ATP binding sites of kinases in different ways, right, to get divergent effects in terms of outputs. And if you look at that example, you'd say, well, of course, that makes sense. You have a kinase that's coupled to an RNase. You have an enzymatic activity coupled to the kinase. So, how general can this concept be? And what I'll tell you is most kinases do not have RNases, right? So what about for the other kinases that don't have enzymatic activities coupled, which is the vast majority of the kinome? So I'm going to tell you a very fast story about this. And I'm going to give the credit where it's due in terms of who did this work. This is Ames Register work, as well as Sujata Chakraborty, Emily Dieter, and Linglan. And what they worked on is trying to understand for kinases that serve scaffolding roles within the cell, can we actually divergently modulate their functions to their ATP binding site? So just a quick backstory here. We spent many years studying a family of kinases called the Src family kinases. In many cases, you learn about Src family kinases in an intro cell biology class. They're a good example of our understanding of how you can regulate the kinase activity of a kinase through multiple domains. How these kinases work is they can exist in an autoinhibited form that has low catalytic activity. Release of this autoinhibited form activates the kinase domain and promotes their ability to phosphorylate substrate. What we showed over a number of studies, a number of biochemical studies, is that by stabilizing two different conformations that I showed you at the beginning of my talk with ATP competitive inhibitors, we could either promote the closed form of Src family kinases or the open form, which involved either the release of these regulatory domains or the engagement. And so what we knew is we had this ability to modulate these regulatory interactions which play a role in scaffolding through the ATP binding site. And what we were really interested in though, is if we did this, would this actually make a difference phenotypically? Would the ability to open up those regulatory domains and promote intramolecular interactions in a cell actually influence the role that Src plays? And based upon what we know about its scaffolding role, there's reason to believe so, but we really didn't have the tools to answer that. So the main question here is, as I showed you before, for Src, would adding or treating cells with two different inhibitors that stabilize different conformations give you divergent phenotypic effects? So unlike IRE1, where we spent a number of years and we've been lucky in terms of having really good reagents for testing this that have high selectivity, for the Src family kinases, we never really had anything that would allow us to do this, And we didn't want to spend years developing small molecules that would be very selective, but basically stabilize different ATP binding site conformations. So we used sort of a trick that was developed by my postdoctoral mentor, Kayvon Shokat, and we try to use this idea of rather than taking and developing a molecule that is highly selective for your kinase of interest, is can we actually use the ability to introduce mutations that will sensitize a kinase to an inhibitor to basically do a similar study. And so we wanted, in this case now, to sensitize Src family kinases, but other kinases as well, to these conformation selective inhibitors, so inhibitors that would stabilize different conformations, and then use these reagents, in a chemical-genetic system to, in this case, stabilize Src in either one of these active site conformations I showed you. We tried many iterations of this. So we tried bump hole strategies, we tried different complementation, and the thing that Ling Lan that worked out that actually ended up being the key is we developed this, we have a cute acronym for called systematic, but rather than using a bump hole or some other complementation strategy, we actually use a strategy where we introduce a cysteine that is unique within the ATP binding site of a kinase, which will then sensitize it to an ATP competitive inhibitor that contains an electrophile. And so in this case, this is just in vitro inhibition data where we have Src kinase either with our sensitizing cysteine mutation, which is shown in color. We can see all three inhibitors. This stabilizes the active form. This is the alpha helix out, and this is the DFG out. This mutation sensitizes Src to all three of these inhibitors, but in black is shown the wild type as well as, I'll show you, the majority of the kinome using our kinome profiling method are resistant to this inhibitor. So what we have now is the ability to introduce a single mutation, which we've shown through enzymatic assays have a minimal effect on the catalytic activity of Src and other kinases. And by introducing mutation, we can sensitize it to the conformation selective inhibitor of our choice. All right, so we set out to do this with Src. We have a cell line that makes this a fairly easy assay. These are mouse fibroblasts that are deficient in Src family kinases, so Src, yes, and Fin. We took these cells, we introduced a GFP fusion of Src that, I'm not showing the wild type data, but we have the wild type versus the phi to C. We see the similar localization. We can then take our cells that are expressing the sensitized version and either add our inhibitor that stabilizes the conformation where the regulatory domains are intramolecularly engaged, or in this case, our inhibitor that is selective for the sensitized version of Sark that causes the regulatory domains to disengage. And the first thing we saw when we did this is Sark rapidly relocalized to endosomes. All right, so that was one sort of phenotype. So we could affect the localization of Sark depending upon what type of ATP competitive inhibitor is bound to. That doesn't necessarily mean function. So the next thing we saw was probably one of the most dramatic results I've had in my whole career. First, I'll show you something that is not dramatic, and that's where we add our inhibitor that stabilizes the intramolecular engagement, so diminishes the scaffolding function or prevents the scaffolding function of Sark. I sped this up, not much happens. It looks like a fibroblast. But now if we compare this to our inhibitor in this case that releases the regulatory domains of Sark. Here we go. Here's what we see. So here first we just see some filopodia formation that's sort of normal for fibroblasts, but within 5 to 10 minutes we see these blebs form, right? And so this is not after a day, this is not after 12 hours, this is within 15 minutes of adding this inhibitor. So we see redistribution of Sark to endosomes and the formation of these blebs. And this will go on and on and on. We can stop imaging so we don't photobleach it and continue. And we see this cycling and this blebbing that occurs. So most people know blebbing from apoptosis, right? It's a normal blebbing of the cell membrane. Well, first, before I show that, I'll just say we can do this. I just showed you a movie. We can do the same assay. We can treat with inhibitors. We can score the cells. And what we see is that only in cells that contain our sensitized Sark mutant do we see this blebbing. If we have the wild type version treated with the same inhibitor, which is resistant, we don't. If we have our other inhibitors with a sensitized Sark kinase, we don't see it. So it's very specific to the sensitized kinase in the presence of this inhibitor. All right, so what is cell blebbing? This is a form of non-apoptotic cell blebbing. And what it is a version of three-dimensional cell motility, right? And I don't have time to go into this today, but what we were able to track down and show that by stabilizing Sark in this conformation, we're able to promote activation of Rho GTPase, which leads to retraction of the actin cytoskeleton, which the overall hydrostatic pressure within the plasma membrane causes the plasma membrane to actually form these blebs, which then are pulled back in by the actin cytoskeleton. And so basically we were able to promote in an inhibited kinase by releasing its regulatory domains a phenotype that is consistent with increased motility. All right, I'm going a bit long. If you're interested in this, we have some details in these papers. What we were able to then track down is what promoted this function overall. As I mentioned, it's a Rho GTPase-driven process, but in doing so, we actually uncovered a role for this N-terminal lipid binding domain of Sark that showing that it plays a regulatory role that also is able to, when released with this inhibitor, promote this blood formation. This is work that we performed with our collaborators in genome sciences, Doug Fowler and Ethan Aller. But it basically just shows, again, how, in this case, we can sort of use our ability to modulate the ATP binding site of kinases to discover new things. All right. So I told you 2 stories today. As I showed you, that's 2. We can do math, right? I showed you 2 kinases. There's 538, so we have 536 to go. But we think this general concept is, or we think this concept is general. As we learn more and more about the multifunctional roles that kinases play, and we learn more about the allosteric relationship between the kinase domain and these other domains, we think that some of these methods and tools we're developing will basically give us the ability to sort of broadly explore this allosteric modulation. And what we hope it does tell us is 2 things. One is just from a basic standpoint, science standpoint, right? How are these kinases regulated? How do the domains talk to each other? And then from a drug discovery standpoint, can we validate not only which kinases are the most beneficial to actually modulate with a drug, but how do we want to modulate them? And can we, in some cases, for kinases that have been targeted in the past and maybe have not been successful in the clinic, maybe we can, by better understanding how we can modulate them, we can basically take another swing and say, well, maybe we need to reevaluate not just our ability to turn off their ability to phosphorylate their substrates, but the other functions they play in the cell. All right. Finally, I'd like to thank all the people. I did my best to try to acknowledge all the people that did this work. As I mentioned, Hannah and Feroz, and I briefly mentioned Likun, were instrumental in this IRU1 alpha work. I try to also acknowledge Ames, Sujata, Emily, and Lingland for their really fantastic work, and the Stark family kinases, which I didn't have time to tell you, to give it its full credit that it's due. And then, as I mentioned, I have a lot of great collaborators like Feroz, as well as collaborators at UW, which have allowed us to both sort of move in new directions and also sort of establish some of these very exciting platforms. Finally, I would like to acknowledge my funding, and finally, thank all of you for your attention, and I'd be happy to answer any questions.
Thanks, Dusty, for that really great talk. Are there questions for Dusty?
Great talk. I thought that was really cool. One of the things I thought that was really cool was just your steering between sort of the XPP1 splicing, what I like the kind of classic IRE1 activity, versus RID, which I think is kind of more like the shredding the RNAs that are proximal to the ER. And you had these compounds that could kind of shift you between the two. Is it that XPP1 has those extra sequences in its RNA that enables it to still be spliced with very low RNase activity, and that's sort of more like the RID pathway is blocked just because there's not that much RNase activity, but there is enough to support the specific XPP1? Or is there something more about how an XPP1 splicing occurs versus the regular RNA?
Yeah, no, that's a great question. So our model is that it's the first explanation you mentioned, right? That essentially we're dialing down, right, the activity of IRE1, and if you do that, right, you basically end up getting the preferred substrate over where you need more elevated activity to act upon the messenger RNAs. And we know this too, even from, if we take Akira, which completely inhibits, but we basically can, drive down, basically get partial occupancy, we can actually see a similar effect, right?
So yeah, if you titrate the Cura drug to a lower amount,
yeah,
do the same sort of steering.
Yeah, so, and I guess that's also too, right, in terms of motivation, we had a feeling, right, if we were able to invest in developing these compounds, that it would actually work because we knew that was the case. But from a drug discovery standpoint, right, it's not, and I would say even in some of these in vivo models, the reason we've been able to use Curas and they've worked is because we can dose them, right, at sort of like this partial inhibition, but as a therapeutic, it's not really possible to do that, right? You want to basically be able to, just in terms of heterogeneity and everything else in terms of blood levels, you need to say, shoot for a target range, and usually it's going to be maximum occupancy.
So that's why it's valuable for the drug when it's saturated to only have a partial.
Exactly.
Even though you partially inhibit with
Yeah.
That's
Other questions?
Is there any known downside to inhibiting the terminal UPR in terms of either immunity or cancer?
Yeah, so, I think, so immunity, I think there's, as far as we can tell, right, there's a pretty minimal effect on the immune system. I think the most negative thing you could think of IRE1 would be the adaptive response, because in terms of B cell differentiation, you need to actually build out the ER, and IRE1 is a big component of that. So that would be in completely suppressing IRE1 something. And we actually show with our molecules that we can take Curas versus these pairs, these partial antagonists, and the pairs allow B cell differentiation. In terms of the terminal, I don't think there's much. I mean, there's at least as far as we know that no downside. There's not really a downside to inhibiting the terminal UPR. So that's, and then in terms of cancer, yeah, we just don't know yet, right? I think, there are quite a few people working and actually trying to, especially for B cell derived cancers and things that have a upregulated UPR, and actually trying to develop similar molecule like curas for cancer therapeutics. But I think in terms of whether these, have a higher propensity for cancer, it's unclear. Yeah.
It was an awesome talk. This is way out of my area of expertise, so I'll qualify it this way. But do you know if the mechanisms you're suggesting, the multiple conformations for the binding site and the effect that it has on like multimerization is true for other nucleotide derivative binding proteins, like proteins that bind GTP or the cyclic nucleotides?
Yeah, that's a good question. I think I was actually to Scott about this before. I think in theory that it should be held, right? Like any enzyme, right, that uses allosteric to modulate active site residues, right? If that's the case, we should be able to identify molecules that will stabilize the conformations, right? That the movement of those residues, and that should have a bidirectional relationship. I think the thing you run into is that most, especially GTPases, ATPases, they're really hard to develop small molecule inhibitors of. And so kinases are kind of funny molecules. They bind to ATP, right, a highly polar molecule, but for reasons that no one knows, there's no known reason for, they like, the main binding determinant is through the adenosine, so the hydrophobic part, and not the phosphates. The problem with a lot of ATPases and GTPases is what they really like are the phosphates. And so in general, I think we haven't been able to really test a lot of these things because we just don't have the molecules to test it. I guess you could come up, it would be interesting to try to make, triphosphate analogs and things that could potentially do this. But I think there's just less motivation because it would all be a sort of biochemical endeavor. Yeah.
Hey, why don't we thank Dusty again for a really great talk? Before everyone stops you, I'm sure millions of people are going to be up here in minutes. I was planning to meet you at 5:45
Okay.
at the hotel for dinner. Does that make sense?
Yeah, that's great.
Okay.
Yeah.
And I think Jason's going to...
Really great.
Oh, thanks.
It was really fun talking with you.
Yeah.